Let's turn our attention to a couple of things relating to enzymes. And one is enzymes that catalyze multi-substrate reactions. It's very easy to think of an enzyme going, okay, A goes to B. But what if the enzyme has A plus B goes to C plus D? Multiple substrates that the enzyme can act on. Well, so what, you might think? What's the big deal? Well, the big deal is there's different ways that these enzymes can catalyze that reaction. Okay? So let me explain a couple of these mechanisms for you. The first of this is easy to understand. It's called ordered. An example of an enzyme that does this is pyruvate dehydrogenase. We'll talk about this enzyme later in the term. Not pyruvate, I'm sorry, lactate dehydrogenase. Sorry, I'm naming it by the wrong side. Lactate dehydrogenase. Okay? This enzyme has what's called ordered binding. And what ordered binding means is that the enzyme will bind one substrate first before it binds the other one. And I don't know the particular order this one binds. I'm guessing it's pyruvate first, and then it binds NADH. And it will never bind NADH first. And there's something that you've learned already this term that you already know is consistent with this. That is weird today. What is that? Not cooperativity, no. Okay, conformation of the enzyme. And what specifically am I referring to?
Well, one substrate has to bind the enzyme in order for the enzyme to change shape in order for the other substrate to.
Okay, so this is consistent with the Koechlin-induced fit. The binding of the first substrate causes the enzyme to change a little bit, and that's required for the binding of the second substrate, which is what you're saying. Yeah. This ordered binding is completely consistent with the Koechlin-induced fit. Binding of one is required for the binding of the other. That means that's very, very good evidence that the enzyme has to change in some way upon binding its substrate. This tells us that the Fisher lock and key model is not a perfect model for all enzymes. In fact, as we shall see, it's not a very good model at all for enzymes. But the Koechlin-induced fit model is consistent with ordered binding. Okay? Well, how about if we have a situation where we have non-ordered binding? And by the way, don't worry about these schematics. I find they're more confusing than anything else. Okay? Don't worry about the schematics. Other enzymes have what are called random binding. They bind multiple substrates, but it doesn't seem to matter which one they bind first. This enzyme, creatine kinase, can bind creatine first, or it can bind ATP first, and it really doesn't seem to matter. Does this mean that the Koechlin-induced fit model is wrong? No? Okay, well, that's good. My heart was stopping there for a second. Okay. It doesn't mean it's wrong. Why? What's that? Well, either one could change the shape. In fact, both of them probably change the shape. And they both probably change the shape, but the shape that they're changing has nothing to do with the binding of the other one. There's a lot of regions in an enzyme that can change shape that have nothing to do with substrate binding. So random binding means that when we see those changes of shape that happen in the Koechlin-induced fit, that the changes of shape simply aren't affecting the binding site for the other substrate. So it doesn't matter which one binds first. Okay? Okay. Question? Yeah. I'm sorry? Does it mean that the domains are farther or closer apart? Actually, it has very little relationship with that because changes that happen on binding can be moved far distances away. So it's a good question, but the answer is no. It doesn't really have anything to do with distance. Okay. Well, there's one other type of binding that I want to talk about, and students tend to find it kind of confusing. You guys have been a good group, so hopefully you won't find it so confusing. This one is a mechanism referred to as double displacement. And I like to refer, and a lot of people like to refer to this mechanism as ping pong. A ping pong table, the ball goes over there, the ball comes back over here, the ball goes over there, the ball comes back over here, right? And it's kind of boring to watch, all right? But the ball is going from One side to the other side to the other side to the other side. In the ping-pong mechanism of catalysis, okay, or the ping-pong mechanism of binding, if you want to think about it that way, the ping-pong mechanism of binding of substrate, what's happening is the enzyme is actually changing as it goes through its reactions. How does that occur? Okay, here's a reaction called a transamination reaction. We'll talk about transamination reactions later. In a transamination reaction, what's happening is the amine group, which is right here on aspartate, is being swapped with an oxygen group on another molecule. So this amine is going over here. That puts the oxygen over here where that amine was, and it puts that amine where that oxygen was. Now, it's very easy to think switching. We just go, and it switches. But the reality is that that's kind of simplistic. In reality, that amine has to be carried by something over to this molecule. It has to drop off that amine and grab the oxygen and put it back over here. This is the ping pong. On the ping side, this guy is grabbing the amine. The amine is physically attached to the enzyme. We have made a covalent intermediate of the enzyme, and the covalent intermediate has the enzyme attached to the amine group. So in this ping part of the reaction mechanism, the enzyme is physically linked to the amine group. It's pulled it off of the aspartate. The enzyme in the ping mode goes over to the other side of the table where it's ready to become pong, and in pong it drops off the amine and grabs the oxygen. So now the enzyme is bound to oxygen. It's been ponged. It now goes back over here and deposits it back over in this place. Ping, pong, ping, pong. This swapping back and forth of the enzyme between one state and another, the ping state having the amine, the pong state having the oxygen, gives us two different enzymatic intermediates, and that's why we call it ping-pong mechanism. Transamination reactions are very important in amino acid metabolism, meaning that we are making and interchanging amino acids. Here's aspartic acid right here. We've converted aspartic acid into oxaloacetate, which is not an amino acid, but we've converted alpha-ketoglutarate, which is not an amino acid, into glutamate. So in essence, we've created glutamate and we've lost aspartate. That's really useful for a cell if a cell has too much aspartate sitting around, and it needs glutamate. Transamination reactions are very common among amino acids, and that swapping back and forth allows cells to make the proper amount of each amino acid that they need. Okay, questions about that? Yes? Good question. So if I were to ask you on an exam, I would think the best questions come out of the students. I should take you guys questions, just write your exams from that. But if I take an enzyme and I put an amine on, do you suppose I'm going to change the enzyme? I am. So I'm going to make a slight shape change, and when I put the oxygen on, I make a slight shape change again. Will that affect the enzyme's activity? It might or it might not. But any kind of covalent modification I do to an enzyme is going to have at least a slight shape change on that enzyme. Good question. Okay. All right, so much for binding and so forth. Other questions? Let's see. Before I talk about inhibition, I want to just show you kinetics for what are referred to as allosteric enzymes. And I've used this term before. When we talked about hemoglobin, I said hemoglobin exhibited cooperativity. And cooperativity meant that the binding of one molecule by a protein changed the affinity of binding of that protein for the same molecule, additional units. And I said allosterism is the term we use to describe that in enzymes, and it is. Okay? Allosterism occurs in the same way. The binding of the first substrate or the first molecule that the enzyme binds changes the affinity of the enzyme for other molecules of the same type. This requires multiple subunits, just like cooperativity requires multiple subunits. The units have to interact with each other, and the binding of one unit affects the other units. And we'll talk more about this next week when we talk about mechanisms of catalysis. But not surprisingly, excuse me, we see when we plot the velocity of the reaction against the substrate concentration that we see that sigmoidal curve that we saw with cooperativity. Pure indication that you've got what's called allosterism. Now, allosterism is a very, very important concept. I'm going to say a lot more about it in the rest of the term. But suffice it to say that allosterism provides cells with a way of controlling enzymes once they've been made. That race car going to Fred Meyer, you'd like to be able to put the brakes on every now and then? Allosterism allows you to put the brakes on in certain conditions. You're getting ready for class and you have to get there and you're late and you need to speed up. Allosterism provides an enzyme a way to speed a reaction up when it needs to. So allosterism is a very important concept. As I said, I'll talk about it next week. But it allows the cell to have some control over what an enzyme is doing. Okay. Let's turn our attention to enzyme inhibition, because enzyme inhibition is really where it's at when we start thinking about drugs. People design and companies design pharmaceuticals to knock out bacteria, to knock out viruses. And what they're basically doing with these pharmaceuticals, for the most part, is designing molecules that are inhibiting critical enzymes that a cell needs to have. So if we understand something about how inhibition of enzymes occurs, we understand a great deal about how it is that drugs are designed. Okay? Now, there are two main types of inhibition I'm going to talk about, and I meant to delete this one that says uncompetitive. I'm not going to hold you responsible for uncompetitive. You may delete that because I think you'll find it very confusing, and it's not necessary for our purposes for you to understand that. So you may delete uncompetitive. What I'm going to be talking about in here are competitive inhibition and non-competitive inhibition. Okay? Well, what do these different terms refer to? Let's talk about competitive inhibition first. Okay? Competitive inhibition occurs when I have a molecule that looks like the normal molecule that the enzyme binds. That is, it looks like a normal substrate, but it's not exactly the same. Dihydrofolate is an intermediate in the synthesis of nucleotides. It's necessary to make nucleotides. There's an enzyme that catalyzes the metabolism of, or catalyzes the reactions of folate, dihydrofolate. It's called dihydrofolate reductase. You don't need to know that. We'll talk about it later. That enzyme is necessary for cells to divide. Because if cells can't make folate, they can't make nucleotides. And if they can't make nucleotides, they can't divide. So if I inhibit that enzyme, I stop that cell's ability to divide. If that cell is a cancer cell, I've got an anti-cancer drug. And that's exactly what you see on the bottom. Methotrexate is a molecule that looks an awful lot like dihydrofolate, but it's not dihydrofolate. When dihydrofolate reductase, the enzyme binds onto methotrexate, nothing happens. It just sits there. Okay? Now, if I am a cancer cell and I've got methotrexate, I am not going to be able to be a cancer cell anymore. I'm going to die. Okay? Very, very useful thing. Now, this type of inhibition I've been describing to you is called competitive inhibition. What you have are things that are competing for the active site of the enzyme. And the reason that they're competing is that they have the same basic structure. They both will fit into the active site. This means that when we have a molecule that is a competitive inhibitor of the enzyme, a competitive inhibitor of an enzyme will always look like the normal substrate of the enzyme. You could look at these two on an exam and say, whoa, they look alike. We're talking about competitive inhibition. Now, one of the things that students frequently overlook when they think about competitive inhibition is that example I just gave you is, oh, it's very simple. Yeah, that's great. And the way you're going to think about it is, that dihydrofolate reductase is going to grab a hold of that methotrexate, and it's not going to let go of it. Okay, that would be a mistake. Competitive inhibitors do not involve covalent bonds. They do not involve covalent bonds. That means that there's an on, off, on, off. And the concentration of the inhibitor will be critical. The more inhibitor I have, the more likely it's going to be on instead of dihydrofolate. Now you might say, well, why don't we design something that just knocks out the enzyme? Well, we'll talk about some things that knock out enzymes, but in general, such molecules are pretty nasty. Methotrexate, for example, if I give this to a cancer patient, the cancer patient usually gets quite sick. You know, people who get some types of chemotherapy get quite ill, and the reason that they get quite ill is because their normal cells are getting the drug as well as the cancer cells. And if I were to completely knock out the dihydrofolate reductase in all of the cells, the organism would die. I cured the cancer, but the patient didn't survive. Not a good career move, okay? So, competitive inhibitors do not covalently bond to that enzyme. So what I do when I give this treatment to patients is I give them this for a period of time, and then I give the patient a bunch of this. And the reason being that for that period of time that I've given the patient this, cancer cells are dividing more rapidly, and therefore they're going to run out of folate quicker, and they're going to die quicker. Some of the normal cells are going to die. But I try to rescue those cells as much as I can, as quickly as I can, by giving folate. If I had covalently bound all of my dihydrofolate reductase with this material, it wouldn't matter how much folate I provided back to the enzyme. Okay, everybody understand that? Yes, Sean. How long would you wait? You know, I'm not a physician, I don't know, but on the order of hours. It wouldn't be a long time that you would wait to give this. No, not necessarily. It would depend on the type. There's many types of cancer, different division rates and so forth. Yes, Phil.
So you're giving them, let's say, in order to kill the cancer cells, and then you're trying to save you're gonna
The normal
save
cells.
the normal cell by
That's the hope, yeah. Okay. Yes, Amber. Oh, good question. Is there a difference in enzyme affinity between the two different molecules? What do you suppose? A no? No's? Actually, if you see any difference, you would expect that you might see a little bit change in affinity. And I don't know in this particular case, but in most cases, you probably will see a little bit of change of affinity. Yes. Yes, Lisa, right? Oh, yeah. Can you get rid of cancer only using this? In general, probably not, no. Because this is a sledgehammer way of killing things. And so this might help to reduce the tumor size. This might help to get the body's immune system back on track. But this is a sledgehammer. This is a tough way to do it. And this is used for other things besides cancer. But it's used in very small doses. OK. All right, so you've seen competitive inhibition. I'm getting better at that. Okay, you've seen competitive inhibition. What in the world then is non-competitive inhibition? Actually, before I do that, let's talk about the kinetics. Okay, kinetics. This is a confusing graph. Let's look at two things. Let's look at the black line and look at the top green line and ignore the others. All these are different concentrations of inhibitors, and we're not going to be focusing on different concentrations of inhibitors. If I have an enzyme that's a competitive inhibitor, the enzyme is competing with the substrate for the active site of the enzyme. When I do my inhibition kinetics, I'm going to plot velocity of reaction against substrate concentration, right? I want one variable. What's the one variable I'm going to have in this experiment? Substrate concentration. So I've got one set of tubes that have a fixed amount of inhibitor. The other set of tubes have no inhibitor. And each corresponding tube has the same substrate concentration. When I do this experiment, I discover something interesting. As I go to ever increasing amounts of substrate, notice when I start out that the inhibitor is lower than the uninhibited. That's no surprise. We would expect that an inhibitor would inhibit the enzyme, right? So if I have an inhibitor in here, I'm going to be knocking out the ability of the enzyme to function. So it's lower, but as I get higher and higher amounts of substrate, what happens, these two lines, by the time they get over here, pretty much overlap with each other. Why did they do that? Why is it that the inhibition disappears as I get increasing amounts of substrate? Because competition's going on. There's more substrate to outcompete the inhibitor. Let's imagine over here on the left side that I've got 100 molecules of substrate and I've got 10 molecules of inhibitor. As I increase substrate concentration, I get over here where I've got 1,000 to the same 10. And over here I've got 1,000,000 to the same 10. Eventually those 10 are going to disappear from the equation because the substrate is going to basically blind the enzyme to the inhibitor. It's not going to see it. It's going to be 99.99% more likely to find substrate than it's going to be to find inhibitor. That tells us, first of all, that when we have competitive inhibition, Vmax does not change because of that competition. Now, how about Km? What do you suppose happens to Km? Same? Different? What's that? Roughly the same? That's kind of copying out, Jose. Phil? For which one? Why would it be larger for the inhibitor? Okay, so going back to the boyfriend-girlfriend, you're walking down the street, right? And there's a lot of members of the opposite sex that are very attractive, right? One of you is going to get distracted, right, by what's going by, and the other one, right? You've been there? Your attention is not going to be totally devoted to the person that you're standing next to. Should be. Let's not say it shouldn't be. But if it's not, right? Okay, you're not going to be devoting all that attention because there's other distractions. The other distractions are the inhibitor. It's going to take more substrate. You're going to have to talk to that person a lot more so they pay attention to you when that happens, right? It's going to take more substrate in the inhibited reaction to get to Vmax. Vmax over 2 to get to the same amount of attention. It's going to take more substrate to get to Vmax over 2. Make sense? So, in competitive inhibition, Vmax is unchanged. Km is increased. Okay. Now, I've got enough time, I think, to introduce non-competitive inhibition. And what I'm going to do is that I'm not going to show you, there's another kind of graph I'm not going to show you. I'll show you that at the beginning of the next period, and that'll be the end of the material for the final exam. So that'll take about maybe 5 minutes maximum at the beginning of the next period. But before I do that, I want to talk about non-competitive inhibition. Okay. Now, in the case of competitive inhibition, we schematically saw that as this. The inhibitor looked very much like the normal substrate. Non-competitive inhibition, first of all, tells us from its name, the inhibitor is not competing with the substrate. The inhibitor is not competing with the substrate. This means that the binding site for the inhibitor on the enzyme is not the active site. It's not the active site. And the binding of the inhibitor affects the binding of the substrate. Does this sound like Kochlin's induced fit to you? I hope so. In this case, the induced fit is coming from a non-active site. There's something on the outside of the enzyme that this inhibitor is binding to. It changes the enzyme such that, hey, the enzyme no longer recognizes that substrate. Somebody has just come and stolen your boyfriend or girlfriend away. Okay, now, that's what's going on in the case of non-competitive inhibition. And what happens as a result of that is an exact flip-flop of what we saw going on with competitive. Let's think about this. When I had competitive inhibition, I had 100 molecules of substrate to start, and I had 10 molecules of inhibitor to start. I was 10 times more likely to have substrate find the enzyme under those conditions, right? I could think of it, that concentration of substrate and inhibitor, about 1/10 of my enzyme is going to be inhibited at that time. And by the way, these are reversible. Come off, come on, come off. But it comes off here, it goes over there. So on average, about 10% of my enzyme in the competitive inhibition is going to be inhibited when I have 100 to 10. When I have 1,000,000 to 10, 99.9% of the enzyme is going to be active. So the amount of enzyme that's active is a function of the concentration of the substrate. In non-competitive inhibition, that's not the case. No matter how much substrate I add, I can't make any more enzyme be active. The amount of enzyme that's going to be inactive is going to be a function solely of the amount of inhibitor I add. So if I start out with 10% of the enzymes inhibited, it doesn't matter if I add 1,000,000 times more substrate, I'm still going to lose 10% of my enzyme's activity. I want you to think about this. If I take and do two sets of reactions, one set of reactions where I have 100 enzyme molecules, and I take another reaction and no inhibitor, and I take 90 enzyme molecules, which one's going to have a higher Vmax? Come on, you guys know this. The 100. Because Vmax depends on the amount of enzyme. In the case of the inhibitor, I knocked out 10 of those enzymes. So that tells us that when we have a non-competitive inhibitor, Vmax falls. It does not fall when we have a competitive inhibitor. With non-competitive inhibition, Vmax decreases. Now I'm going to tantalize you with something that will puzzle you the heck out of you, but it's true, and that is that non-competitive inhibition, the Km stays the same. Km stays the same for non-competitive inhibition. I may briefly mention that at the beginning of the period next time, but in non-competitive inhibition, Km stays the same. Okay, what I want to do today is finish up a very tiny section on enzymes, and then that'll be the last of the material that you'll be responsible for on the exam. The exam is written. I think it covers the material reasonably well, and it definitely is in exactly the same format that you've already seen on the practice exam. Okay, well, let's look at a Lineweaver-Burke plot when we have a situation where we have a competitor and competitive inhibitor. And I think one of the things that this is another situation where I think students sort of memorize figures and without really looking to see what the information on the graph is telling them. So I'm going to try to tell you what's on this graph and how you can determine this graph. Okay? So a Lineweaver-Burke plot, you remember, plots reciprocals of the data and the information on the axes, 1 over velocity versus 1 over s. A couple of you have asked me about your book where it uses 1 over v0. What is v0? And v0 simply is another way of saying that you do the velocity very quickly. 0 stands for initial velocity. So when I told you do these reactions for a couple of seconds, that's what you're doing, a v0. Okay, so you can call it v, you can call it v0. It's the same thing in the way we're using those terms. Now, what I showed you before is if I take that hyperbolic plot that I got for a v versus s plot and plotted it, I saw, in fact, that I got a straight line. And I told you that the intercept of the x-axis was minus 1 over km. And the intercept of the y-axis was 1 over Vmax. Now, if I tell you I take another set of reactions, and instead of having no inhibitor, I add an inhibitor, what would I expect to see? Well, you can look at this and you can memorize this graph, but I really don't want you to memorize this graph. I want you to use your own knowledge to do it. Let's think about how you might do this. First of all, if I do competitive inhibition reaction, what do I see? I see what happens to Vmax. Vmax stays the same. What happens to Km? It changes, and how does it change? It increases, okay? So that information is enough to sketch out this plot right here. If I know that I've got a line that goes here for an uninhibited reaction, and I know that the Vmax is the same, I know that the line for the competitive inhibitor has to cross at exactly that point. Didn't have to memorize that. I simply had to know that the Vmax didn't change. And when the Vmax doesn't change, 1 over Vmax does not change either. I also know that the Km increases, and when the Km increases, minus 1 over Km gets closer to 0. And that's what happens here. So now I know 2 points, that point and that point. And guess what? 2 points define a line. I now know the line that the competitive inhibitor would have. Okay? So I can do that without memorizing lines and curves and shapes and that sort of thing. And I actually use the knowledge that I have about these enzymatic reactions to do that. Make sense? Okay. All right. Well, let's look at the same thing for a non-competitive reaction. If I do a non-competitive reaction, I do a V versus S plot, the first thing I see is this. The Km changes. I'm sorry, the Vmax changes. Here's the Vmax for the no inhibitor. And if you focus your attention just on this top line right here, you see that, whoa, we've got a different Vmax. The Vmax coming in at about maybe 45, whereas up here the Vmax is coming up at about 95. Okay? So this tells us that the Vmax is changing, the Vmax is decreasing, and you understood that last time, I hope. Okay? The other thing you remember from what I said last time is that non-competitive inhibition, the Km does not change. And I didn't tell you why, and I've decided I'm not going to tell you why. I'm not going to hold you responsible for knowing why, but if you want to know, I've already had a few people ask me, come talk to me. I can give it to you in a couple of minutes that I'm not going to spend here in class on why the Km does not change. Okay? But in non-competitive inhibition, the Km does not change. You don't have to worry about this E, the S, E plus I, blah, 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 blah. I hate those things myself. Now, if we do a Lineweaver-Burke plot for the no inhibitor versus the non-competitive inhibitor, we see this sort of a plot. And again, we can use the same sort of knowledge that we had to make this determination. First of all, we remember that when we have non-competitive inhibition, the Km does not change. Therefore, minus 1 over Km will not change. The two lines are at that point. One of the confusing things that students overlook or they forget on an exam is the fact that the line for the non-competitive inhibition is higher, and this is where I know people are trying to memorize lines instead of memorizing principles, is higher than here. Why is the line higher? Because, well, think about this. For example, I said that the Vmax goes down. Why is the line higher? Yeah, the reciprocal of something going down is something going up. So the reciprocal of a Vmax going down is a Vmax going up. Okay? So 1 over V, that line, that point is right there. Knowing those 2 points now, I know the line and I know what the shape of the plot is for a non-competitive inhibited reaction. Okay, so questions on this? Clear as mud? Okay. Let's see. There's only a couple other brief things that I'll say, and then I'll finish the material for this exam. One is an inhibitor. We'll talk actually a little bit more about this on the material for the second exam. And this is everything you've seen so far that is competitive and non-competitive inhibition. These types of inhibition are reversible, meaning that the inhibitor binds to the enzyme, but it doesn't bind covalently. There are things that will bind to enzymes, and when they bind to enzymes covalently, they basically kill the enzyme because that covalent bond is too difficult to break. The first of these is a very general kind of inhibitor. It doesn't have a specific name, and it's a general kind of inhibitor because it's reactive for a specific group. The group that this inhibitor is specific for is the hydroxyl R group of serine. The inhibitor is called DIPF, which has a long name that I won't give you here. DIPF, okay, makes that covalent bond, and if serine is in the active site of an enzyme, you've just killed this enzyme. If it's not in the active site of the enzyme, you may or may not kill the enzyme. It depends on the effects that it has on the structure of the protein. But if it's in the active site of the enzyme, that enzyme is dead. So one of the ways that we can see if a serine is in the active site of an enzyme is to treat the enzyme with DIPF and see, does it lose its activity? If it doesn't lose its activity, we may assume that that serine is not in the active site of the enzyme. Now, this type of inhibitor is a relatively small molecule. The enzyme doesn't recognize it. It's small enough that it fits into the enzyme just fine without any other kind of alterations that need to happen to get it in there. The enzyme has no affinity for it as such because it doesn't bind. This is a small molecule that squeezes its way in. I'm going to show you in a second an example of where the enzyme actually binds to an inhibitor that then covalently binds to the enzyme. And that type of inhibition has a name, and I'm going to show it to you. Questions on this before I go to that, though? Okay, so DIPF, a very general type of inhibitor specific for serines. We can do the same thing with iodocetamide, with cysteines. Okay, very general. Again, it sort of squeezes in. The enzyme doesn't have any affinity. If we have a cysteine in the active site, iodocetamide will bind to it and inactivate the enzyme. So again, we have a test to see if a cysteine is in the active site of an enzyme. Now, I told you that if the enzyme has some affinity for, that is, it binds and recognizes and binds to a molecule, and then the molecule makes a covalent bond with the enzyme, we've done an inhibition that is an irreversible inhibition. And by the way, DIPF and iodocetamide do irreversible inhibition. They make covalent bonds, the enzyme is dead. But this type of inhibition I'm talking about is that not only are we making a covalent bond with the active site, but the molecule that's binding at the active site looks like the normal substrate. So the enzyme sees this molecule, it grabs a hold of this molecule, and this molecule goes, ha ha ha, got you, and then makes a covalent bond with the active site, and the enzyme is dead. This type of an inhibitor is called a suicide inhibitor, because the enzyme commits suicide in binding to it. Now, the example I have on the screen is not a very good one, unfortunately. There's a better one. And the better one is exactly how the antibiotic penicillin works. Penicillin works by binding to an enzyme in bacteria that's necessary for making the bacterial cell wall. And penicillin looks like the normal substrate that this enzyme recognizes and catalyzes a reaction on. But when penicillin binds to it, it covalently links itself to the enzyme, and the enzyme is dead. Okay? So, suicide inhibition has a relationship with competitive inhibition. The relationship being that they both involve inhibitors that look like the normal substrate. They differ in that competitive inhibition is a reversible inhibition, whereas suicide inhibitors are irreversible. They make covalent bonds. Okay, and there are other examples, including the one on the screen, but like as I say, it's not a very good example, unfortunately. Okay, if there are no questions, that's where I will stop, actually. The material